All right, so what we talked about, I'm going to just show you the notes because we did go along with the notes when I was doing those drawings. It's just that I wasn't, I was drawing more than I was following them. Sometimes I'm going to actually pull up the notes and I'll say, okay, this is what it says. So, we talked about the different divisions of the nervous system, but we used a chart, all right? So I will be doing this a little bit more. We talked about the resting membrane potential. We talked about depolarization and the graded potential. So what we want to talk about to start. Okay, we're going to talk about continuous propagation. We ended talking about the refractory periods. And the absolute refractory period we said was when you cannot get another action potential because either you're already depolarizing, so another action potential is not going to happen, or the inactivation gate is closed, so you're not going to get another action potential. So that's absolute. You cannot get another action potential no matter what. And then we said that the refractory, relative refractory period started because the inactivation gate opened, but the activation gate was closed, right? So now you're going to get a stimulus, but it's going to depend on how strong of a stimulus do you need. If the cell is closer to threshold already, you're not going to need much of a stimulus, right? If you're already at 62 millivolts, you only have 2 millivolts to go to reach threshold. But if your cell is at negative 90 millivolts, now you've got 30 millivolts to go, so you'd need a much bigger stimulus to even reach threshold. We're going to talk a little bit about that today, all right? So before we do, just to talk about continuous propagation, and I did not, and the second one down here is saltatory. It's not written in there, but it's saltatory. So we were talking just about the initial segment. We said all of those positive charges funneled into the axon hillock, and then the current took them into the initial segment, and it's at the initial segment where you had to get that negative 60 millivolts in order for those sodium voltage-gated channels to open. And as soon as that happened, the entire action potential occurred. And so once you hit threshold, an action potential is going to occur in that initial segment no matter what. That's called the all or none principle. If you hit threshold, you're going to get an action potential no matter what. So that's all or none, okay? But now, the only thing that's depolarized is that small area, that initial segment, right? That's the only thing that has depolarized. So that was meaning it's here where all of the sodium gates opened up, and then the sodium gates closed, and then where all the potassium gates opened, and then where all the potassium gates closed to create our action potential. It all occurred right there. And so these positive charges that are moving into the initial segment as the sodium is bringing them in, they're going to get carried by the current down to the next segment. So they get carried on down to the next segment of the axon, and they're going to also reach threshold just because there's so many sodium moving in and positive charges moving down. That next segment is then going to reach threshold too. It's going to go from resting up to negative 60, and boom, you get another action potential. Sodium is going to move into the cell, it's going to get carried down by a current, and then the next segment is going to depolarize. And so in this type of what we call, this is called propagation, and it is the movement of the, it's the movement of the action potentials down the axon. We get an action potential here, then we get an action potential here, then we get an action potential here, okay? It's continuous because every segment, every adjacent segment has to depolarize in order to have an action potential. We had this one, then this one, then this one. So segment 1, which is the initial segment, then segment 2, and then segment 3. each one of them, they each have to depolarize. So we're moving down the line, right? So in the game of hot potato, we're taking a potato and we're passing it to this guy, then to this guy, then to this guy, and we're passing it down the line, right? Well, there's some other neurons that have that myelin that surround it. And myelin is created by a cell called the Schwann cell in the peripheral nervous system. And then in the central nervous system, it's called the oligodendrocyte that produces it. But this is like a peripheral nervous system neuron. And so this nucleus is producing all of this myelin, and it surrounds this area of the neuron, and we call that an internode, right? It's called the internode, where that myelin is surrounding it. Well, it's a fatty substance. The myelin is fat, right? So sodium cannot get in. It can't get in through there. It can't get anywhere near those sodium and then the potassium. There's the gates. It doesn't matter what's going on. The gates aren't going to respond because, I mean, even if they were open, the ions can't get through because of the fat. So what happens here is that the positive charges of sodium, they're still carried by the current, but they're going to get carried down to a little segment of axon that's not covered by the myelin. And that we call the node, where it's not covered by myelin. That's called a node. We used to call it the node of Ranvier, but we kind of like to get rid of the scientist's name. So now it's just, the node and the internode. Let me see if on your lab list if it still says, it still says node of Ranvier, so you still have to put it in there. Node of Ranvier, all right? So anyway, this is the next place where sodium can enter then. The voltage-gated channel's open, sodium comes in, we depolarize, we get an action potential, okay? Then we have the internode again, and sodium cannot get into, it can't go anywhere near those gates. But the positive charges keep moving down. Here's another node, and now sodium can get in, right? And then we have another internode. So when we look at this, and we look at our hot potato game, here we have, the hot potato, it's going to get passed way down here, and then way down here, and then way down here. So it's like the people are standing a lot farther apart. This is going to be a lot faster in saltatory, right? Because they're passing it down instead of passing it just to the next person, right? So saltatory propagation occurs on a myelinated axon, and it's faster. That make sense? Okay. So that being said, here's a picture. You're going to see these. These are on your lab list. So you have to identify the cell body. You have to identify the dendrites. You have to identify the myelinated axon. So on the model, this here is really the myelinated axon. But I think you actually see it. I don't know where it's tagged, but it's not tagged where where it's tagged, it looks like it's actually on the myelin itself. But that's where, so make sure you're looking at that. That's where I'll put the sticker, okay? And then you have to, then the axon terminal is on the lab list. It's either axon terminal or synaptic terminal. Don't put both. Don't ever put any dashes or slashes or anything like that. Just put one or the other. Doesn't matter. You'll get it right. This is the myelin sheath. And then the node of Ranvier is right in between those little segments that have no myelin on them. Okay? And then here we see on this picture, this is an unmyelinated axon, and we do have unmyelinated axons in the peripheral nervous system. They're part of the autonomic nervous system. We're going to touch base on that when we get to chapter 16. Okay? What? Oh, bless you. Okay. This is a good review. It kind of shows you where everything happens. So This doesn't look very bright, does it?
No. It's hard to see.
Yeah, it's hard to see. It's better than the document camera. Let me see. Whoop. Let's see how I can. Whoop. Shoot. Okay. Okay. So now what I want to start with is I want to look at, after that nerve impulse gets all the way down to the synaptic terminal, what's going to happen down there? Well, we're going to see the release of a neurotransmitter, and we're going to talk about acetylcholine, that's a cholinergic synapse, and we're going to see what happens there. How does acetylcholine actually get released? How does it bind to the sodium chemical gated channels on this cell, right? So when we're talking about this, you're going to hear the terms presynaptic cell and postsynaptic cell. The cell that's sending the message is the presynaptic cell. The cell that's receiving the message is the postsynaptic cell, right? So we've got presynaptic and postsynaptic cell. So let's look at what happens when that nerve impulse reaches the end. So this is, see in your drawings, I'm going to show you this first, and then we're going to go to your drawing, okay? So this is the presynaptic cell. This is the postsynaptic cell. Here we see this red line. That means an action potential is coming down to the axon terminal, and it hits the axon terminal, right? So the first thing that's going to happen is calcium-gated channels are going to open up. Right? So this is just showing the action potential getting there. Okay? So when the action potential gets there, calcium channels open up. Well, calcium is higher in concentration in the extracellular fluid than it is in the intracellular fluid. So based on diffusion, calcium wants to go in which direction? It wants to go into the cell. So calcium goes into the cell. It's that calcium going into the cell that causes these vesicles to fuse with the cell membrane and release acetylcholine, release the neurotransmitter. Now, where did we get those vesicles from? Where did that all come from? Where did we get the neurotransmitter inside here? Where did I say it was made?
The nucleus?
Yeah, the nucleus neurotransmitters are a protein. So the nucleus is making those, making the acetylcholine, and the acetylcholine is traveling down to the axon terminal in these little vesicles. And each little vesicle contains about 3,000 acetylcholine molecules. So that's quite a bit. So calcium enters, and those vesicles fuse, and they release acetylcholine out into this area that we call the synaptic cleft. The synaptic cleft is it's the space between the presynaptic and postsynaptic cell. Okay? So that's what we're seeing is acetylcholine being released. The next step then is we see the acetylcholine goes to the postsynaptic membrane, and it binds to the channels on the postsynaptic membrane. It binds to those channels. Now those are chemical-gated sodium channels. Okay, chemical-gated sodium channels. So they bind to those chemical-gated sodium channels. We know what happens. We know the story from here, right? The channels open up, sodium floods in, the cell depolarizes, and we get our action potential, our very first action potential, if it's strong enough, right? If there's enough acetylcholine and enough sodium coming in, we get our first action potential. So we know the story from there, right? Now, after that, there is a neurotransmitter, not a neurotransmitter, there is a chemical that are these little yellow-shaped, moon-shaped things. It's an enzyme that's called acetylcholinesterase. Acetylcholinesterase. And its job, it's always just floating around in that area. Its job is to bind to the acetylcholine, pull it off of the receptors, and then break it down. And it breaks it down into an acetyl and a choline group. Okay? And then that ends, that's going to end then that transmission of the nerve impulse, right? As long as a nerve impulse is coming down, acetylcholine will continually be released. So when is that? Like when you're exercising, your muscles need this constant innervation from your nerve. So nerve impulses are constantly coming down, and then acetylcholine is constantly being released, and a new action potential is constantly being produced on the postsynaptic cell. In that case, the postsynaptic cell would be the muscle, and it would cause the muscle to contract, right? It's only going to stop when you stop exercising. Then the nerve impulses stop. Then ACH stops getting released, okay? So if we go here, this is just showing you the 4 different, the steps here. It's showing how there's an action potential that comes down. It's showing how when it comes, so this is the first step where you just need an action potential. So action potential arriving is the first step. The second step then is that action potential causes calcium gates to open up, so calcium comes into the cell, and that causes acetylcholine to get released into the synaptic cleft. The third step then is that the acetylcholine travels all the way across that synaptic cleft and binds to the chemical gated channel, the sodium chemical gated channel. And now sodium can influx or move into the cell. And we know what happens. If there's enough sodium moving into the cell, we get depolarization. Right? We get depolarization and an action potential if there's enough, if it reaches threshold. Okay? And then the last step is that this acetylcholinesterase is going to bind to the acetylcholine. It's going to take it off of there, and it's going to split that acetylcholine back into an acetate molecule and a choline molecule. And the choline molecule will be reabsorbed back into that axon terminal, and it can be reused to create more acetylcholine. So now we have 2 sources of acetylcholine. We have the cell body, the nucleus making it, putting it in the vesicles, and it travels down the axon. And now we also recycle it. So we have 2 ways of filling up those synaptic terminals. Okay? 2 ways of filling up the synaptic terminals in that axon. Filling up the synaptic vesicles in the axon terminal, right? Okay, so a couple of things. And now that's kind of, you should probably have heard of that, right, in general A&P. There's a couple of terms that I want you to know about. First of all, there is a term called synaptic fatigue. Synaptic fatigue. And the other one is a synaptic delay. So let's talk about the synaptic delay first, right? Synaptic delay. So in the synaptic delay, we have, what does a delay mean? Delay means it's not going very fast, right? It's something slowing it down. There's a delay. So the synaptic delay occurs between the time calcium diffuses into the cell and crosses over to the postsynaptic membrane, right? So that it all takes time. It takes time for calcium to diffuse in. Then it takes time for acetylcholine to move across the synaptic cleft. That all takes time. That's a delay. That's a normal delay, right? Well, in your nervous system, sometimes you only have 2 neurons involved. So now you only have 1 synapse, and that's what this is called. This is called a synapse. So if you only have 1 synapse, your delay is going to be a little bit shorter, right? Because you only have 1 synapse, 1 delay, right? In this 1 delay, by the time a second action potential releases acetylcholine and you get another action potential on a postsynaptic cell, the first action potential has already gone down that postsynaptic cell 3 centimeters, right, in 1 delay. The first action potential's moved 3 centimeters already, right? So the delay is pretty significant. Now, if you had let's think about fatigue. Fatigue is when the cell runs out of acetylcholine. Okay? The cell runs out of acetylcholine. When's that going to happen? Well, it's going to happen when you have exerted yourself too much. All the vesicles here will be exhausted, right? And the cell itself can't make acetylcholine fast enough, and the choline that's in the acetylcholine that's being broken down in the synaptic cleft, it can't be reabsorbed fast enough and reused fast enough. So that is when your neurons become fatigued. They've run out of acetylcholine, right? Because again, it takes time for the nucleus to make new acetylcholine and for the old acetylcholine to be reabsorbed and reused. And when we bring things back into the synaptic terminal here, we call that reuptake, right? It's a reuptake. We're going to see that a little bit later when and you've heard that probably before, like a serotonin reuptake, right? Medication, right? SSRI. So you'll hear that term again, that reuptake, okay? All right. Now, say if you just have 1 synapse, you only have 1 delay right there, right? But if you have a pathway where you have more than 1, right? And that first action potential has traveled 3 centimeters down, that first action potential could be right here before you even get the second action potential going, right? So the more neurons you have involved, the greater the delay, basically, right? Does that make sense? Okay. Fatigue. I want to make sure I put these in order in this one. Okay. So we're going to look at these notes a little bit here. We have other neurotransmitters in the body, okay? Acetylcholine is one that we start out with because it's kind of a simple one. It has a direct effect it binds to the receptor, opens the ion channel, and the ion moves in or moves in, okay? So this is, it's kind of easy. But neurotransmitters, like acetylcholine, can be excitatory, and it can cause that postsynaptic cell to depolarize, like we've been seeing. But in other places in the brain, if acetylcholine binds to a receptor, it can be inhibitory, and it can make threshold go further away. It can make the resting membrane potential go further away from threshold. Okay, so when we go closer to threshold, that's excitatory. If a neurotransmitter makes you go closer to the threshold, it's excitatory. If a neurotransmitter makes you go further away, that's called inhibitory. All right? And acetylcholine is found in many areas in your brain, and so when it is excitatory, so basically what I'm saying is, yeah, it's the neurotransmitter that has to be released, but it's really the property of the receptor. What's the receptor going to do with that message? Is it going to open the gate, or is it going to keep the gate closed? So there's different receptors in your body, and it's really the receptor, not the neurotransmitter, that determines the response of the postsynaptic cell. Okay? So keep that in mind. It's the receptor, not the neurotransmitter, that determines what the response is of the postsynaptic cell. So there are 4 different types of neurotransmitters, 4 different classes, basically. And so we have biogenic amines, amino acids, neuropeptides, which are also called neuromodulators, and we have dissolved gases. So we're going to take a look at each one of those. The biogenic amines include norepinephrine, dopamine, and serotonin. So first I'm just going to go over those, and it's just something you have to memorize what they do. At the end, I'm actually going to show you what a couple of these, how they actually work on the neurons, okay? So, so norepinephrine, that's the first one we're going to talk about. You've heard about this one before. Norepinephrine, we said acetylcholine is found at cholinergic synapses. Norepinephrine is found at adrenergic synapses. So whenever you hear adrenergic, you automatically think, okay, that's norepinephrine, right? That's adrenergic. And it's always going to be excitatory, okay? This one is not like acetylcholine. Norepinephrine is always excitatory. It's always going to depolarize that postsynaptic cell. It's basically going to mobilize your brain for action, right? Norepinephrine in the central nervous system, it's going to cause your brain to go into action. Then we have dopamine, and dopamine's also found in many areas of the brain. Dopamine can be excitatory or inhibitory depending on the receptor, okay? So if it's excitatory, it causes a high. Dopamine causes a high. And what cocaine does? Cocaine inhibits the removal of dopamine. So dopamine stays on the receptor, and that's where the high comes from with cocaine, okay? Dopamine can also be inhibitory, and we're going to see this with like Parkinson's. So dopamine, it inhibits acetylcholine. And so acetylcholine is stimulatory, makes muscles really twitchy, right? And we'll see this later, but I'm just going to explain it really quick. Acetylcholine, if left on its own, would cause you to be constantly having tremors. Dopamine is released from an area in the brain, and it inhibits acetylcholine so that you don't tremor, so that you can have smooth movements and you're not tremoring. So in Parkinson's, dopamine is gone. You're not producing the dopamine, so the acetylcholine is just having its way with the muscles, and they're all twitchy, okay? You get lots of tremors. So that's dopamine. That's how it's inhibitory. And then finally, we have serotonin. Serotonin is also found in the brain, and it increases attention span, and it also increases your sense of well-being, right? Well-being. Helps to regulate sleep. So there's an antidepressant called an SSRI, or a selective serotonin reuptake inhibitor. All right, so let me get this right there. All right, so if we had our neuron here, okay, and the neuron was releasing serotonin, and then serotonin made you feel good, got rid of the depression, right? But it's going to be broken down. So then serotonin gets broken down, and it gets brought back into the synaptic terminal so it can get reused, right? So what a selective serotonin reuptake inhibitor does, long word, SSRI, is that it inhibits the reuptake of serotonin. So now serotonin accumulates in that synaptic cleft, and it can keep binding to the receptor, and you can keep feeling good. Does that make sense? It's a reuptake inhibitor. It doesn't allow the reuptake. So serotonin stays bound to the receptor, leading to wellness. All right, then we have Gaba. Gaba is another, so Gaba is no longer our biogenic amine. Okay, it's an amino acid. There's two amino acids really that I want to talk about. GABA's the only one that's on here. I'm going to tell you about another one though. GABA is the main inhibitory transmitter of the CNS. Okay, so it's inhibitory and it's in the CNS. It's the main one in the brain. Okay, so it's like Valium. That's what it is. It inhibits the brain. It slows the brain down. Person has vertigo and they feel constant motion, then Valium, just like GABA, Valium binds to the same receptors that GABA would. It causes things to slow down and inhibit and relax, right? So that's the main neurotransmitter in the brain, main inhibitory neurotransmitter in the brain. There's another amino acid one. So again, these are amino acid. Another amino acid one is called glycine. Glycine is an amino acid. This is the main inhibitory one in the spinal cord. So this will be another one that you'll sometimes hear about, right? So GABA is in the brain, and glycine is the spinal cord and the brain stem. They're both inhibitory. Both of them are inhibitory, okay? Okay, then we look at these neurotransmitters that are called neuromodulators. So what they are is they're basically going to alter the rate of how fast that neurotransmitter can be released from the presynaptic cell. So they're influencing the presynaptic cell, right? So here's our presynaptic cell, postsynaptic cell. The presynaptic cell has to release the neurotransmitter. A neuromodulator would be released from another neuron and cause that neurotransmitter to be released even faster. Okay, or slower. I mean, that's what a neuromodulator does. It has an influence in how fast the presynaptic cell releases the neurotransmitter. Okay, so okay. All right, so now I want to just briefly go over, acetylcholine is not our only neurotransmitter, right? So if we look back here, I'm just going to go back to, all right. So, go back to this picture here. And let's make a new picture. Okay. So we have your, here's your postsynaptic cell. So there's your postsynaptic neuron, the cell body, right? And then we have these presynaptic neurons. Right? And like I said, some of them are going to release a chemical that opens sodium channels. So sodium can go in. Okay. And some of them are going to release a chemical that will open potassium channels so that potassium will move out. And then they become summated. Like, what's the net effect, basically? Like, basic math. What's the net effect? You have some that stimulate, some that inhibit. What's the net effect? So if you have, and this, when sodium is coming in like that, this neurotransmitter, this action potential, it's not even an action potential yet, it's a graded potential. This graded potential is called an excitatory post-synaptic potential, or an EPSP. This one over here, where potassium moving out, that's inhibitory because it's making the cell more negative, taking it even lower than -70. That one is an inhibitory post-synaptic potential, okay? And the summation is just what's the net effect. If you have more EPSPs, the cell is excited. If you have more IPSPs, then the cell is inhibited, and it's going to take an even bigger response to bring it up to threshold. Okay? Now, when you have a cell that is receiving more EPSPs than IPSPs, we say that that cell is facilitated. Okay? And we call that process facilitation. Facilitation. And so facilitated means that the neurons just moved closer to threshold. It's no longer sitting at -70, it's now up a ways. Maybe it's at -65. Because there's been impulses that have been facilitating that postsynaptic cell. It's been bringing it close, even though it hasn't reached an action potential, hasn't reached threshold yet, it's bringing it closer. So we say that neuron's facilitated. Okay? So if it's highly facilitated, that means it's really close to threshold. That means it's probably close to -61. That's a highly facilitated neuron because there was enough sodium coming in that it would take a very small stimulus to cause an action potential to occur. Okay? So, for example, nicotine stimulates the cardiovascular centers in the brain and the respiratory centers in the brain. It stimulates them. It brings them closer to threshold, right? That's what nicotine does. So it needs very little stimuli to reach threshold and cause the heart to beat faster and the lungs to breathe faster because it's so facilitated. Right? So maybe just walking down the hallway after a person's smoked or vaped, right? Their heart's going to beat faster, and their lungs are going to breathe faster, and it wouldn't be like that if they hadn't had the nicotine. So that's what nicotine kind of facilitates a neuron. Okay, so now last thing that we're going to talk about. Instead of talking about what we're going to do is now we're going to look at influences on the presynaptic cell. So I know, just to complicate this just a little bit more, because there's two drugs that we want to talk about. You're going to hear about these in pharmacology, right? So we want to talk about two drugs that influence that presynaptic cell. Okay? So how can the presynaptic cell be affected? Well, we have the, actually I think I have a picture here. We're going to look at Gaba, and we're going to look at serotonin, and we're going to see what did we say Gaba was? Did we say Gaba was stimulatory or inhibitory? So Gaba is the main inhibitory neuron. So this is what Gaba does. Gaba is released from a neuron, and it affects the presynaptic cell. Gaba inhibits the calcium channels, okay? So it's going to inhibit some, but not all. It inhibits the calcium channels. So not as much calcium is going to go into that presynaptic cell. If you have less calcium diffusing in, you have less neurotransmitter being moved out through exocytosis. So it's inhibiting, in effect, this presynaptic inhibition. So this is presynaptic inhibition because we are inhibiting the presynaptic cell. It will also inhibit the postsynaptic cell. Okay? So we got a lot of neurons here. We've got this neuron and this neuron. So when GABA is released from this neuron's called the presynaptic cell, but it gets released here and it blocks calcium channels on this neuron, less calcium is going to come in. Now this cell becomes the presynaptic neuron, and so if less calcium is in here, less neurotransmitter is going to be released, and we're not going to get that stimulatory effect. It's going to be an inhibitory effect. That's how GABA inhibits. It actually affects the presynaptic cell, not the postsynaptic cell. Okay? So that's one example. And the other one is that serotonin, and I already kind of told you about serotonin. Serotonin also affects the presynaptic cell. Okay? Serotonin actually opens those calcium gates. So more calcium comes in, which means more neurotransmitter is going to be released. So serotonin is then an excitatory. It has an excitatory effect on the presynaptic cell. So we have to term these things. Serotonin causes presynaptic excitation because it's causing the presynaptic cell to become more excitable. Okay. And GABA, we would say, is presynaptic inhibition. Because it's blocking the calcium channels on that presynaptic neuron, so less calcium is coming in and less neurotransmitter is going out, right? So that's what presynaptic inhibition and postsynaptic inhibition is. So I would ask you maybe, how does serotonin cause presynaptic excitation? And you would just say, well, it excites and opens more of the calcium channels. So then more calcium comes in and more neurotransmitter goes out. Or how does GABA affect the presynaptic neuron? Okay, so it's not less memorization and it's more just understanding, right?